Hello guys, so welcome back to the channel Plant Pathology Pathshala. So I am Dr. Jhumishree Meher, PhD Plant Pathology from Banaras Hindu University. So as you all know that we are doing a series on plant diseases. So first we are covering the rice diseases, and in rice diseases we have already covered rice blast, BLB, then rice tungro disease, and another the rice brown spot. Okay, so four diseases we have already covered. Everything about the disease you can get in that video. So okay, separate videos are there, you can check out that. So in this video particularly we are going to discuss about sheath blight of rice. So similar three sections will be there. Section 1, the MCQ based question. Section 2 will contain the Assertion reasoning type questions, and at the end in the section 3 we will discuss quick facts for quick revision. Okay, there you will get a short version notes on the sheath blight of rice. This is also a very important disease of the rice. So let's start the video. First, the causal organism of sheath blight of rice is Rhizoctonia solani. This is a fungi that is causing sheath blight of rice, and its sexual stage is Thanatophorus cucumeris. Okay, so let's start the section 1. Question number 1. Sexual stage of sheath blight pathogen is. I have already answered this question, so here the answer is option number B, Thanatophorus cucumeris. Okay, next, question number 2. Which environmental condition is most favorable for sheath blight development? Option A, 15 to 20 degrees Celsius with 50% RH, means relative humidity. Option B, 20 to 25 degrees Celsius with 70% RH. Option C, 30 to 32 degrees Celsius with 96 to 97% RH. And option D, 35 to 40 degrees Celsius with 40% RH. So here the answer is option C, 30 to 32 degrees Celsius with 96 to 97% relative humidity. So the if you if means in short, this disease, the sheath blight of rice, it favored by high temperature as well as high humidity, RH. Both temperature and RH should be high enough for the development of sheath blight of rice, that is 30 to 32 degrees Celsius and 90 to 96% RH. Next, question number 3. The characteristic initial symptom of sheath blight is. Option A, spindle shaped lesion. Option B, greenish to grey oval lesion near water level. Option C, black lesions on corn. And option D, smut balls. Here the answer is greenish to grey oval lesions near water level. So this is this leaf. This is the stem area. In this, so leaf will be formed here. So what happened in the sheath blight symptom actually appear on the leaf sheath around the stem. Okay, so there oblong to elliptical spots will be developed that are generally greenish to grayish in color. Later it will changes this, and the central portion will become grayish and with a brownish margin, which ultimately affect the upper leaf blade also, and entire sheath will become blighted. So initial characteristic symptom is greenish oval lesion near water level. Spindle shaped lesion, this is characteristic for the blast, rice blast. Okay, next, question number 4. The most important survival structure of the pathogen is, means for the Rhizoctonia solani. Option A, oospore. Option B, chlamydospore. Option C, sclerotium. Option D, teleospore. Here the answer is sclerotium. So the Rhizoctonia solani produce sclerotium or sclerotia, which is helpful for the overwintering or oversummering of the pathogen. Next, question number 5. Excessive nitrogen application increase seed blight severity because option A, it kills antagonist. Option B, it creates dense humid canopy. Option C, it reduces inoculum. Option D, it inhibits sclerosia. So here the answer is it creates dense humid canopy. Because of excessive application of nitrogen fertilizer, what happened? Excessive vegetative growth will be there, that leading to the dense humid canopy that favors seed blight infection. Next, question number 6. The pathogen causing seed blight belongs to. So the options are option A, Ascomycota, option B, Basidiomycota, option C, Oomycota, and option D, Mucoromycota. So here the answer is Basidiomycota. Initially Rhizoctonia solani, this was first initially grouped under the Deuteromycetes fungi, but as per the latest classification of Kerketel 2008, this Deuteromycete, this group was deleted. So the fungi, they are then placed either under Asco and some are under the Basidiomycetes. So now this seed blight pathogen Rhizoctonia solani was placed under the Basidiomycota. Next, question number 7. Which stage of rice is most susceptible for seed blight infection? So the option are option A, seedling, option B, tillering to heading, option C, dub stage, option D, booting stage. So here the answer is tillering to heading stage. This is the most susceptible stage for the seed blight infection. Next, question number 8. A major reason for the importance of seed blight is that the pathogen is. So option A is vector borne. Option B, it has more than 188 host species. Option C is seed transmitted only. Option D, infects grain only. Here the answer is it has more than 188 host species. This is a wide host range pathogen. That's why it is very important. That is reason making it a major disease or major important disease of rice. In MCQ based question also it was asked that dash is a wide host range pathogen. Rhizoctonia, then Magnapothe. Option will be there. You can choose. You have to choose the Rhizoctonia option. Next, question number 9. Movement of sclerotia through irrigation water contributes mainly to option A, primary infection, option B, secondary spread within the field, option C, seed infection, and option D, vector transmission. So the answer is option B, secondary spread within the field. So through the irrigation water, the sclerosia spread within the field and causing the secondary spread of the disease. Then question number 10. Which biocontrol agent is widely recommended against seed blight? Option A, Rhizobium. Option B, Azospirillum. Option C, Pseudomonas fluorescens. Option D, Frankia. Here the answer is Pseudomonas fluorescens. Next, question number 11. The disease triangle component most responsible for epidemic in dense rice strand is option A, host nutrition, option B, microclimate, then option C, soil texture, and option D, seed size. So here the answer is option B, microclimate. As we have already discussed that the epidemic are generally caused due to the dense canopy development due to excessive application of nitrogenous fertilizer, that actually making this, that changing the microclimate, making it more humid. That's why the pathogen is able to multiply more easily. Okay. Next, question number 12. Which practice is least desirable under seed blight prone condition? Option A, wider spacing. Option B, balanced fertilizer. Option C, excess nitrogen application. And option D, field sanitation. Here the answer is option C. Why? Because other rest three, these actually used for the management of seed blight, but excess nitrogen actually favors the disease. Next, question number 13. Thanatophorus cucumeris is classified under, we have already discussed, this was classified under the Basidiomycetes fungi. Option A is correct. Then question number 14. The lesion expansion pattern of seed blight is best described as option A, circular spots only. Option B, coalescing lesions covering large seed area. Option C, when limiting streaks. Option D, rot posture. Here the answer is coalescing lesions covering large seed area. Wherever blight is written means initially the spots will be smaller only, but later it coalesces to cover the entire surface and causing the blighted appearance. Next, question number 15. The most suitable long-term management approach obviously for the seed blight is option A, fungicides only. Option C, resistant variety use. Then option D, integrated disease management. Option D, flooding alone. So here the answer is integrated disease management. Why it is not the resistant variety? See, for the wide host range crops like Rhizoctonia, no such resistance genes are available. So this is not an option. We can go for integrated disease management for long-term sustainable approach for the management of seed blight pathogen. Next, section 2, Assertion reasoning type question. So Assertion 1 is the seed blight is more severe in dense planted rice field. Reason, dense canopy maintain high humidity around the seed. Yeah, so both option A and both A and R is true, and R is correct explanation of A. Next, assertion 2, excess nitrogen favors seed blight. Yeah, this is true. The reason is luxuriant growth creates favorable microclimate. So both A and R are correct. Option A is correct, okay? Because the excess nitrogen, it is modifying the microclimate in favor of the seed blight. Next, question number 3, assertion. Sclerotia is important in epidemiology. Reason, they survive in soil and crop residue between the season. Yeah, both the statement, both are correct. Both A and R are correct, and R is true explanation for the A. Next, question number 4. Seed blight is considered as a monocyclic disease. Yeah, this is a monocyclic disease. Then reason, multiple cycle of infection occur during the season. Wrong. Here it should be single cycle of infection occur during the season. Next, question number 5, assertion. Biological control is useful against seed blight. Option C, reason, antagonist microorganisms suppress pathogen activity. Yeah, both are correct. Next, section C, quick revision points. So, the pathogen infecting the seed blight is the Rhizoctonia solani. Its sexual stage is Thanatophorus cucumis, which is a Basidiomycetes fungi. The initial symptom is greenish to oval lesion that will coalesce and causing the blightening of a large seed. Okay. Next, susceptible stage is tillering to heading stage. Favorable temperature 30 to 32 degrees Celsius with high relative humidity 96 to 97%. So it requires high temperature as well as high humidity. The survival structure is sclerotia, which spread by irrigation water causing the secondary spread. So host range is about it is very wide host range, around 188 species it can infect. Then excessive nitrogen increase the severity by modifying the microclimate. It increase the humidity. canopy humidity. Okay. Next, biocontrol agent uses Pseudomonas fluorescens. Then lesions coalesce and cover the seed, causing the blighted appearance. And best management strategy is integrated disease management. So this is all about the sheath blight of rice. I hope it will be helpful for you for preparing various exams. So thank you for listening.